Welcome to The Explainer. Today, we're getting our hands dirty with a fascinating microbe, Paenibacillus amylolyticus. Is this thing just environmental dirt showing up in our cultures, or is it a genuinely dangerous pathogen we need to worry about? We're going to get to the bottom of that question in this deep dive, and just so you know, all the information we're covering comes from a comprehensive review of scientific literature put together through a Gemini deep search.
Okay, let's just jump right into a scenario that I'm sure is familiar to anyone who spent time at the bench. A blood culture flags positive. You whip up a slide, pop it on the scope, and you see this. A rod that's not quite purple, not quite pink. So what's the call? Is it just a contaminant, something from the skin prep that we can ignore? Or is this the clue that's going to crack the whole case for your patient? This is the exact dilemma we face when we run into the genus Paenibacillus.
So here's our game plan. We're going to follow the entire discovery process for this organism. We'll start with that really confusing gram stain, then we'll move into how we actually identify it and explore its completely different clinical presentations. From there, we'll get into the really practical stuff, the challenges of treatment, susceptibility testing, and infection control. And finally, we'll look at what's next.
All right, so let's start right where the action begins for us, at the microscope, looking at an isolate that is just tricky. It defies easy categorization and can absolutely send you down the wrong rabbit hole if you're not paying close attention. You know, this is the kind of gram stain that just makes you second-guess yourself. You're looking at a mix of purple and pink rods, a classic gram variable picture that can easily make you think you're dealing with something else entirely, but you gotta look closer. The real giveaway, the clue that should make you pause and think, is the presence of these endospores. And specifically, you can see they're inside a sporangium that's clearly swollen. That one feature. That's what tells you you're probably dealing with something in the Bacillus or Paenibacillus family.
So, you have a suspicion that you're in the right ballpark, but how do you get from that hunch to a definitive, rock-solid identification? Well, this is where technology has completely changed the game for us.
And this slide, it really lays out the whole story, doesn't it? The old way versus the new way. On the left, you've got the old-school approach, those frustrating biochemical strips that, let's be honest, often led to a misidentification or just dismissing these bugs as diphtheroids. But on the right, that's our reality now. The widespread adoption of MALDI-TOF and 16S rRNA sequencing has been a total game changer, finally letting us unmask the true clinical impact of this genus by giving us accurate, species-level IDs.
You know, the name itself, Paenibacillus, it literally means almost a bacillus. And this taxonomic shift back in 1993, this wasn't just some academic housekeeping. It was a big deal. It was based on 16S sequencing that showed these organisms were genetically very different from the true Bacillus species. And this is a critical point, because it's the reason why we can't just take the clinical standards and the antimicrobial breakpoints we have for Bacillus and slap them onto these guys. It just doesn't work that way.
So now we've correctly identified the organism as Paenibacillus amylolyticus. The critical question, the one that really matters, is what does this result actually mean for the patient? And believe me, the answer is dramatically different depending on who that patient is.
The clinical picture here is a story of two absolute extremes. In adults, infections are pretty rare. They're opportunistic, and they can show up in different ways: bacteremia, wound infections, but honestly, the prognosis is generally good if they get the right treatment. But in neonates, it's a completely different and terrifying story. In newborns, this genus is linked to a distinct and absolutely devastating syndrome called neonatal panibacilliosis, which means sepsis, meningitis, and just catastrophic neurological damage.
And the severity is just stark. Look at this number, 17%. Let that sink in for a second. That's the approximate mortality rate for the neonatal Paenibacilliosis syndrome associated with this genus. For an organism that many of us once dismissed as environmental dirt, that is a horrifying figure for anyone working in a neonatal intensive care unit. And look, while a lot of that scary neonatal data is at the genus level, mostly from Paenibacillus thiaminolyticus, these specific case reports for Paenibacillus amylolyticus itself confirm that it is, without a doubt, a human pathogen. Here we see it causing a nasal abscess in a patient with diabetes, and here it is involved in a polymicrobial bacteremia. This proves it can cause serious invasive disease. Okay, so that brings us to the next big challenge, treatment. We've identified the bug, we understand the risk, but now, how do we advise the clinical team on therapy? Well, this is where we're basically operating without a map.
And here's the crucial point. There are absolutely no official, evidence-based treatment guidelines for Paenibacillus infections. Not from the Dutch Working Party on Antibiotic Policy, or SWAB, not from the IDSA in the States. Nobody has them.
And what that means for us is that management has to rely on extrapolation, on case reports, and most importantly, on the antimicrobial susceptibility data that we generate in the lab.
So, without a specific playbook, what do we do? Well, we have to look at the next best thing. For an adult with sepsis, we'd probably look to general guidelines like these from swab. Maybe a third-gen cephalosporin for a community source, or a broader coverage with something like Piptazo for a hospital-acquired infection. But you have to remember, these are just starting points. The plan is always to de-escalate based on the definitive lab results that we provide.
Now for neonates, the situation is way more urgent and incredibly challenging. The standard empirical cocktails for neonatal sepsis, we're talking ampicillin and gentamicin, are often completely ineffective against Paenibacillus. Vancomycin has also shown pretty poor results. This built-in resistance might be exactly why the organism can cause such severe disease. The case reports out there are starting to point towards carbapenems, like meropenem, which have excellent CNS penetration as a potentially life-saving option.
All right, so this brings us right into our wheelhouse. How do we test this organism, and how do we advise our clinical colleagues on how to contain it? This is the tangible take-home guidance for what we do in the lab and for infection control.
This, right here, is the fundamental problem when it comes to antimicrobial susceptibility testing, or AST. EUCAST has not established clinical breakpoints for any species of Paenibacillus. This creates a massive interpretive gap, and it forces us to use a surrogate-based approach to try and tell clinicians whether a drug is likely to work or not.
So, what's the best practice right now? Well, we test the isolate using our standard methods, like broth microdilution or disk diffusion. But for the interpretation, we have to borrow. We use the EUCAST clinical breakpoints that were established for Bacillus species. Now, it's not perfect, but it's the closest clinically relevant relative we have to work with. The most important step here is to add a comment to the final report, explicitly stating that this interpretation is an extrapolation. You have to be transparent about it.
Now, for infection control, the whole strategy comes down to one key feature: spores. And that means we model our protocols on what we do for Clostridium difficile. Contact precautions are an absolute must, and this is critical. Hand washing with good old soap and water is mandatory to physically remove the spores. Those alcohol-based hand rubs, totally ineffective. And for cleaning the environment, your standard disinfectants aren't going to cut it. You have to use a proven sporicidal agent, something like a bleach-based product.
Okay, so let's wrap this all up. What has this organism taught us, and what are the critical questions that we, as a microbiology community, still need to answer?
The reality is, we're facing some major holes in our knowledge. We need official AST breakpoints, period. We need evidence-based treatment guidelines, especially for neonates. We need to really dig in and understand the virulence factors that allow this bug to cause such horrible disease. And we urgently need to figure out the transmission pathways that are leading to these devastating neonatal infections.
You know, when you step back, the story of Paenibacillus is just this perfect, powerful reminder of how technology like MALDI-TOF really drives discovery. It unmasks these pathogens that were basically hiding in plain sight, often just getting written off as contaminants. And that really challenges all of us to think about our collective next move. When this happens, how do we build the evidence base for these evidence-poor organisms?
We have covered a ton of ground today. If you want to dive deeper into the primary literature and see the studies this was all based on, you can find a complete list of citations in the accompanying notebook document. There's a link for it right in the description. I'd really encourage you to go explore those sources.
Look, our understanding of emerging pathogens like this is really a community project. It's built on shared experience. So if you've ever encountered Paenibacillus in your lab, please share your insights in the comments below. Your feedback and your stories really help all of us learn. And of course, if you found this explainer useful, please subscribe to the channel for more deep dives into the world of clinical microbiology.